All right, in this section, what we're going to talk about is the continuation of what happens in the development of atherosclerosis, especially now that we have a monocyte that's entered the intima of the coronary artery. So in atherosclerosis, how does plaque formation develop in the intima of the coronary artery? So that's what we're going to talk about and finish up in this section is the development of atherosclerosis. So here what we have is the differentiation of the monocyte into macrophages after activation by the MCP1. So just to kind of review what we've talked about so far is we have the LDL that has entered the intima of the coronary artery, and that has become modified from those special oxygen molecules known as free radicals, and that's going to cause the MCP1 to be made by the endothelial layer. And then we talked about how the monocyte, due to the adhesion molecules, E-selectin, will cause rolling, and then VCAM1 and ICAM will cause sticking to allow for the monocyte to enter into the coronary artery. Now the monocyte is into the coronary artery, so what happens to that monocyte? Well, what ends up happening is that modified LDL, through chemical messaging and crosstalk, will cause that monocyte to change into a macrophage. Really important to know that the modified LDL does not become a macrophage or does not taken up by the monocyte to become a macrophage, but rather the modified LDL will promote the differentiation or the changing of the monocyte into a macrophage, and that's done through chemical messaging. And again, this is just the review of what we just talked about. So the question is, so what happens to the newly formed macrophage? Well, the modified LDL will stimulate, or it will induce the macrophage to release cytokines that stimulate adhesion molecule expression in endothelial cells. So how does that happen? How does that work? All right, so I can get this thing to behave. So end up what happening is that now that you have a macrophage, what happens is that macrophage is actually going to produce these things called cytokines. And think of cytokines, they're special proteins that are produced to cause bad things, generally bad things to happen in tissue. So in this case, the macrophage are going to produce TNF alpha, tumor necrosis alpha. So cytokines, tumor necrosis factor alpha, and also interleukin 1. These are two cytokines that are produced by the macrophage, and they will cause the adhesion molecules E-selectin, VCAM1, and ICAM1 to be made by the endothelial layer of the coronary artery intima. So now what happens is you have the rolling and sticking of the monocyte. The monocyte comes in through crosstalk. The modified LDL will cause the monocyte to change in the macrophage. Macrophage makes cytokines. Cytokines makes the adhesion molecules. Monocytes come in. And you can see that essentially steps 3, 4, 5, 6, and 7 will continue in a vicious cycle to accumulate more and more macrophages inside the coronary artery intima. Again, this is just the written part of what we just talked about. Now what happens is the macrophage, in addition to making cytokines, well, what happens is it actually will then form foam cells. Well, how does that happen? Well, the modified LDL is going to be taken up by the macrophage. And if you remember way back when we talked about the modification of the LDL and how it's attracted to scavenger receptors on the macrophage, well, that is what happens. You have a macrophage here. Here's the macrophage, and here's the scavenger receptor. And the modified LDL is going to be taken up or attracted to the scavenger receptor on the macrophage and taken up into it. And over time, those modified LDLs will form larger and larger lipid droplets, and eventually that macrophage will turn into a foam cell. And eventually, over time, that as it accumulates more and more modified LDLs, it'll become a fatty streak. And think of a fatty streak as if you were to put some butter over a piece of bread, That's a kind of the same simulate what's happening in the coronary artery intima. And then what happens over time, if left untreated, it becomes an atheroma, which is again just the buildup of plaque. And so we have here, putting everything together, this is just kind of to reiterate what we talked about, is that we have the LDL has come from our diet, has come into the coronary artery intima. It becomes modified by the special oxygen molecules called free radicals. Those modified LDL will then cause the MCP1 to be made by the endothelial layer. That will help attract or get the monocytes into the cell. Once inside the intima, the modified LDL through crosstalk chemical messaging will cause that monocyte to become a macrophage. Now the macrophage, it's important to know that the macrophage, there might be some macrophages that start producing cytokines, and there might be other macrophages that actually start taking up the modified LDL to become foam cells. So just keep in mind that some macrophages may do one or the other. Or they may start out producing cytokines and then eventually be take up modified LDLs to form foam cells. But in the event, the macrophage is going to then produce cytokines, TNF-alpha and IL-1, those proteins that are responsible for causing the endothelial layer of the intima to make monocytes, or excuse me, to make adhesion molecules, which will attract the monocytes into the coronary artery intima. It's essentially steps 3 through 7 will repeat in order to keep getting monocytes in and cause the macrophages to accumulate. Likewise, you'll have some other macrophages that are going to take up modified LDL through those scavenger receptors, and they're going to form foam cells. Now, the interesting thing about foam cells is they actually produce growth factors that are going to actually attract and produce attract more modified LDLs, so it can grow and get bigger and bigger. So more foam cells beget more foam cells. You also have metalloproteinases. These are special enzymes. Anything that ends in ASES is an ACEs, is an enzyme that tends to break something down. In this particular case, what's happening is that you have, it's starting to break down the endothelial layer, which we'll talk about in the next section. But what happens with these two, you have cell proliferation, which basically means the buildup of foam cells, and then matrix degradation, which is another fancy term for the breakdown of this endothelial layer. But what you should get here is you should get an understanding of how are the development of plaque in the coronary artery intima. So what I would recommend that you do now is to pause the video, and I would recommend that you, or at the conclusion, at least at the conclusion of this section, is to try to put these 10 steps in your own words. You'll be able to practice this on the blackboard. There's some practice where you'll be able to order this in the proper order. And you can do that multiple times until you have a good understanding of it, because it's going to be important that you have a sound understanding of how plaque develops in the coronary artery intima. And again, this is just the wording of what we just talked about. You can see these in your notes. We also have think and review questions. This is the first time you've seen thinking and review questions. And what is important about this is that it'll allow you to start to apply what's happening in the pathway. So it's more, in this class, it's not just understanding the pathway, so understanding steps 1 through the end, but also being able to use it in real life scenarios. And that's what the thinking and review questions will do. So what I would recommend that you do is that after this lesson, or after this section, is that you try to answer these questions. So for example, what would happen to foam cell development, increase or decrease in the intima if you had a decrease in the LDL in the diet? So let's do the first one together, and then you can do the other ones on your own, and then we can check them. So let's say you had a decrease in LDL in the diet. So let's go back up to here. Oh, excuse me, let's go to here. And so, for example, if you had a decrease in LDL in the diet, what's going to happen to foam cell development? Well, you're going to have a decrease in foam cell development. Well, how does that occur? Well, if you have a decrease in LDL in the diet or in your body due to a healthier eating habits, you're going to have less LDL that will come into the cell. into the intima. Less in LDL in the intima means there's going to be less modified LDL. Less modified LDL is going to cause less MCP1 to be produced. Less MCP1 means there's going to be less monocytes entering the cell, the intima. And therefore, steps 3, 4, 5, 6, and 7 would not occur because you have less monocytes, less macrophages would be produced, less cytokines, less adhesion molecules, and therefore steps 3 through 7 would be reduced. If you have less macrophages, you would have less macrophages to take up the modified LDL to form foam cells. So this is important. When I say justify your answer, you need to go beyond just saying increase or decrease, but also tell us how. I will put there, you'll see that there's going to be a video on going over each of these thinking review questions. So take a moment, do the thinking review questions, and once you've had those, you've completed those, play the video about reviewing the thinking review questions and see if you're right. It's important to make sure that you have a good understanding of what's happening beyond just memorizing the pathway, but how to actually use it with increase and decrease. Because this is some of the questions, these are how some of the questions will be worded on the exam. All right, so this concludes this part. Again, I would take some time to review or outline the steps of each of these and putting them in your own words, making sure that you understand them. And then you can check your answers by going on a blackboard and trying to put these steps in the correct order. Make sure you try to do this over and over until you have a good understanding. Once you have a good understanding, then you should go ahead and do the thinking review questions. All right, let's pause now and let's do some review.